Well, thank you very much for that kind introduction. I'm going to talk to you about almost a quarter century of research activity that has ultimately led to the first disease-modifying treatment for Alzheimer's disease. And it's really taken that long, and it's been wonderful to have been able to participate with many other superb scientists in the development of this type of a material. The outline today is that I'm going to talk to you a little bit about what is Alzheimer's disease? How did it get that silly name anyway? We'll talk about the genetics of Alzheimer's disease, at least what we know about it thus far. We'll talk about the development of antibodies against a peptide, a small piece of a protein called the A-beta peptide. It's also called amyloid, although that's not quite an accurate term. And how this can, in fact, cause problems and how we can generate mice that have amyloid in their brains that is very much like what we find in Alzheimer's disease brain. We're going to talk about how antibodies in humans can actually have an effect on amyloid, but produce a problem that we call ARIA. ARIA sounds so nice. This isn't nice. But how we can manage that. And then we're going to talk about how they've been effective in actually eliminating amyloid, slowing the rate of cognitive decline. And they've now been fully approved by the Food and Drug Administration, and Medicare and Medicaid have agreed to start paying for the use of these types of agents in treating Alzheimer's. Later today, Dr. Jaika is going to talk to you about the plans they have here in Kentucky to begin rolling out this new therapy. So it's a really exciting time to be doing work in Alzheimer's disease. So this is the guy, Big Al. Alois Alzheimer started Alzheimer's disease. Actually, he didn't start it. He studied a woman who had it. And her name was August Dieter. And I can tell you her name because HIPAA doesn't apply after you die. I can tell anything about your health history after you passed away. But she actually, in her 50s, started to develop a memory disorder and ultimately would get lost in her neighborhood. And then she started accusing her husband of having an affair, and was ultimately diagnosed as having pre-senile dementia. Because had she been 75, 80, 90 years old, at the time that Alzheimer's studied her, it would have been thought that she just had a normal part of aging that we called senility or hardening of the arteries or something like that. But she was in her 50s, and when she died, Alzheimer decided to look at her brain. He said, there must be something in there. And this is in the early 1900s. And one of the things about so what do you think the biggest risk factor for Alzheimer's is? Do you know? It's too many birthdays. By far, the greatest risk factor for Alzheimer's is aging. And as we all get older and older, we have an increased risk of developing this disease. In fact, for people over the age of 85, as many as 40% have meaningful forms of cognitive impairment. And so this is a huge, huge problem. And right now, we think that 1 in 50 people in Kentucky have some form of dementia. Many of them never receive a formal diagnosis. Your primary care provider may suspect something, but he doesn't necessarily have the expertise to do the diagnosis. And there wasn't much that could be done for you anyway before now. And so it's estimated the direct care for Alzheimer's is $350 billion a year. Now, a lot of this is when the end stage of the disease, you need to go into aggregate housing facilities, what they call a memory care facility, where they can manage patients much better than a simple caregiver in their own home can do this. But this is 1.4% of the gross domestic product of the United States of America is spent on Alzheimer's disease. So 8-9 years ago, NIH had a budget of about $30 billion to do research. 600 million was going to Alzheimer's disease. And over time, we, myself and others, have gone and talked to our congressional representatives and said, look, as the population ages, as the baby boom generation reaches the age of risk, this is going to cripple our country. It's going to cost so much to care for these people if we don't do something now. So what's happened is year by year, we've been convincing Congress to increase the funding for Alzheimer's until now we're almost up to $4 billion a year. Now, cancer, which costs about 30% less than Alzheimer's in terms of medical care costs, still gets about $6 billion, and heart disease is about $5 billion. But we've made it so that now the funding to do research on this disease is matching the size of the problem. And associated with that, we've now got the first drug, but it's only the first. There's going to be many more. And the more progress that we can make, the more likely it is that those of you who are in this room right now will not have to suffer from this disease. And I think we're very close to reaching that kind of a breaking point. Once we've got the first drug, the rest will follow much more quickly. So what did Alzheimer's see? Well, interestingly, in the early 1900s, a high technology was photography. And they had silver salts that they used in photographic development in order to try and develop film. So he took brain sections from August Dieter, and he developed them like they were film. And when he did this, the silver grains attached themselves to these two, to these two unusual features in the brain. So one of them is this material called amyloid, and it makes what's called a plaque. But inside the neurons, there are things that we call tangles. There were twisted filaments inside there, and they're made up out of a protein called tau. And what we now understand about the disease is the amyloid is building up slowly over 10 to 20 years, like cholesterol builds up in your arteries slowly. And then at some point in time, the amyloid causes the tau to start building up as well. And that's when the neurons start to die. And that's when we see the other changes. But we can identify, so the amyloid is kind of the initiating factor, but by itself is relatively innocuous. You might have some decrease in overall cognitive function, but it isn't enough to make it so you no longer can safely live independently, which is what dementia really does to you. So synapses are lost, neurons die, the brain shrinks by a third. It ends up weighing 2/3 of its normal weight at the end stage of Alzheimer's. So the genetics of this disease has really helped us understand a lot about what is cause and what is a side effect of what we see happening inside the brain. One of the keys is actually a condition called Down syndrome. So Down syndrome is people who are born with an extra chromosome. It happens to be a small chromosome, number 21. And they have a condition that interferes with some of their activities of daily living. But almost all of them, when they get into their 40s and 50s, will develop plaque and tangle pathology like Alzheimer's patients do. And they will develop a dementia on top of whatever cognitive disabilities they've had during their lifespans. And it turns out that the gene that makes amyloid is on chromosome 21. And so simply having an extra copy of this chromosome is sufficient to cause Alzheimer's disease. So that's a key element. We now know that amyloid can cause Alzheimer's. We then also have families. They're rare, but they inherit a mutation that causes Alzheimer's disease. So if you had an affected parent in one of these families, 50% of their offspring would carry that gene and would develop Alzheimer's disease. And they develop early in life, and they develop at a very specific age. So if your family has a history of developing dementia at 45 and you get to 50, you probably don't have the gene. And so this is another key. And we find that many of the mutations that are in these families have either changes in the amyloid gene or the genes that actually cut the amyloid protein into little pieces called the presenilins. And those mutations also can cause Alzheimer's disease. So that's been very helpful to us. But there's a number of other genes that increase your risk. It's not a dominant inheritance, but it can increase your overall risk. And one of the major ones is apolipoprotein E. That comes in 3 variants, 2, 3, and 4. I don't know what happened to ApoE1. But if you have 1 copy of E4 allele, you have about a 3-fold increased risk of developing Alzheimer's. If you have 2 copies, 1 from each parent, you have about a 10-fold increased risk of developing Alzheimer's. It is the biggest genetic risk factor for any known disease, that it's a 10-fold increase in risk. It's a very high increased risk. But we found a number of other genes that also increase your risk, and many of these are associated with inflammation that Dr. Tansey was talking to you about earlier. I can't get too close to this microphone. And so we know that inflammation is playing a role in this as well. Finally, there's sporadic Alzheimer's. We know that there are lifestyle things. You want to avoid head trauma, just letting you know. Don't bang your head against the wall unnecessarily. Diabetes, cardiovascular disease. Education is protective, which is actually somewhat interesting as well. So we've got a simple perspective that I'm going to give you on this. My scientific colleagues, they like to have lots of arrows and things, connections, and this I think really summarizes for you what you need to know about Alzheimer's. Amyloid begins building up for some reason. Can be genetic, could just be spontaneous, but once it starts building up, it will continue to increase in your brain. And by itself, for many years, it doesn't really cause much of a problem. But at some point, the brain looks at this amyloid and goes, what the heck is that? And brings in the immune system to try and do something about it. But the immune system had never seen this before in a way that the immune system was prepared to attack it. So it activates certain programs that the immune system would have used during development or to fight off bacterial disease or viral infections. And they don't necessarily properly take care of the amyloid buildup. But this inflammation, we can show, will induce that tau pathology. So these are the 3 major steps, and it's the tau pathology that kills the neurons and destroys the synapses and causes the memory loss. So we've got this sequence of events, and right now we've got drugs that are working here, but we need more, as Dr. Tansey told you here, and then finally, there's also drugs in development to work here as well. So that at different stages, you may want to be taking different medications in the near future. So I'm going to take you back to 1996. In 1996, at the University of South Florida, we took together 2 mice. One mouse had a mutated form of the amyloid precursor protein gene. Another mouse had a mutated form of the presenilin gene. These are mutations that are known in humans to cause Alzheimer's. When we took these 2 mice and we bred them together in our vivarium, right here, you can see the mice were in love. The mouse from the science paper and the mouse from the Nature paper gave us a mouse that we published in Nature Medicine. So it just shows you how true mouse breeding can be. If you use 2 high-ranking journals, you get another high-ranking journal publication. But this is a summary of what we saw. Non-transgenic mice that were born in the litter, they had neither of the 2 genes. They don't have any amyloid in their brain. This is a mouse, half a mouse brain. These are the hippocampus and the cortex and the striatum, brain stem. And the presenilin mouse by itself doesn't really have much. The amyloid mouse has a little bit of amyloid buildup in the brain. These are amyloid plaques. But when you combine these 2, there's a massive elevation in the amount of amyloid you got. So that's telling us that the presenilin mutation and the amyloid precursor protein mutation are really affecting the same thing, and that it's the buildup of amyloid that really is the key element in these genetics. That was a really important insight to have developed. We also found that the more amyloid that we found in the brain, the worse the memory function was in the mice. So here we're looking at the amount of amyloid along this axis in 3 different experiments, and the number of errors the mice make in our maze path. And you can see that there's a pretty good correlation in multiple studies that we have a linkage between amyloid in the brain and the memory impairments the mice have. Now, these are not severe memory impairments. The mouse might forget where it left its car in the parking, but it's still able to eat, drink, and do all the normal activities of daily living that a mouse needs in order to live. So because of this amyloid buildup, a company in San Francisco called Elan decided to try and vaccinate the mice against amyloid. They wanted to activate the immune system. I'm sure you all have had vaccines at one point or another to fight off not an infection, but this protein that builds up inside your brain. And when they did that, they found in their mice that if they vaccinated them before they started to build up the amyloid, it really completely eliminated it. So these are the control mice here, and this brown stuff is the amyloid in their brain. And if you vaccinated them, it really blocked it quite effectively. And you can see here, there's almost none of it, but the control vaccine mice, they had a fair amount of amyloid that would build up. And so that was a very important observation. And our laboratory with our mouse, one of the things we could do that they couldn't do with their mouse is measure memory function. And we said, well, okay, we think this inflammation is an important thing. And when you activate the immune system, you can promote inflammation. So we predicted that if we vaccinated our mice, what would happen is they would have a premature memory loss, that it would make things worse because of the inflammation. But it turned out that wasn't the case. So we tested our mice at an age when we knew that they could have a memory deficit, or before they would develop a memory deficit at 11 months of age. And what we found is everybody could learn, both the vaccinated mice, the mice don't even have amyloid in their brains, the non-transgenic mice, and the ones that got the amyloid vaccine all learned well. So we waited another few months until our mice normally developed the memory deficits. And the blue line is showing you the mice that did not get the vaccine. They got a control vaccine. The black line is the non-transgenic mice, and they can learn quite well. But what we found is the mice that we vaccinated against amyloid ultimately could learn as well as the non-transgenic mice. And we were able to publish this in Nature. If you don't know, the top scientific journals, for some reason, have a one word in their title. Nature, Science, Cell, Neuron. That for some reason, that's what, so you want single word titles in your journal articles. So Elan, using not only their data, but our data and several other groups as well that replicated what they had seen, they decided that let's vaccinate humans. So they started a vaccine study, and they had a phase 1 study. And in the phase 1 study, they found that not many of the people developed very high amounts of antibody when they were vaccinated against amyloid, because there was an older population. And so in between their phase 1 study and their phase 2 study, they changed the formula for the vaccine. And they didn't test it in enough people. So they then tested it in a large study, and they got a problem. And the problem was that the new vaccine formulation was inducing an autoimmune reaction. And instead of just making antibodies, the immune system was attacking the brain of about 6% of the people in this trial. And that created a big problem. So they stopped the trial very early in the process. And one of the patients died shortly after getting vaccinated. And when they looked in their brains, they could see these T cells that were infiltrating into the brain from the periphery, from the immune system, and attacking the brain. And so that really caused us to have caution about what we were doing. And at the same time, there was a group in Zurich. They had about 35 of the cases in this phase 2 trial. And most of the people had gotten 2 or 3 vaccinations already. So many of them had built up antibodies. Some of them didn't build up much in the way of antibodies. They didn't know who had the active vaccine and who had the placebo vaccine. But what they did do is they went and they measured in their blood how much antibody they had against amyloid. And so when they looked at the individuals who had antibodies against amyloid, they found their cognitive decline over the next year was minuscule compared to people who did not have antibodies against the amyloid. And this told us that maybe there's still something we can do with this. So we were at kind of a decision point in 2004. So active immunization causes adverse effects. It also has variable antibody responses. Older individuals do not vaccinate nearly as well as young individuals. Their responses are somewhat impaired. And so it's a problem that you might run into when you've got an older population that you're working with. The inflammation appeared to involve T cell activation, not antibodies. And so we could do something called passive immunization with what are called monoclonal antibodies. So these are antibodies that are grown in a culture dish, and we isolate them and we purify them, and then we can inject them into you. And we didn't quite do this with COVID. They had some that they were trying to do that with, but this is now common in cancer therapy. They've got a lot of drugs that are antibodies against cancer agents. And also in the immune system, we've got a lot of drugs that are antibodies that are against, and they require an infusion. You can't take them as a pill because the antibody is a big protein that gets broken down inside your gut. So we decided to work with a company called Neuroscience to take antibodies that they had generated and test them in our mouse models. And this is Donna Wilcock. Until about 6 months ago, she was here in Sanders Brown. For some reason, many people who have worked in my lab decided to come here. Dan Lee and my Linda Selenitsa are here, and a lot of the other faculty were graduate students in the research groups that I collaborated with at University of South Florida. But we did an experiment that was unusual. Most of the time when we treat mice with a drug to get rid of the amyloid, we treat them before they even started to develop the amyloid. It's a prevention type of treatment. But in this study, we waited until the animals got old. They got to the age of about 18 months and we started treating them, and we stopped after 1, 2, or 3 months of treatment. And because of what we saw, we decided before we publish, we better replicate this. So we took a second batch of mice that we took from 2 years of age out to 28 months of age. Typical mouse lifespan is about 26 to 30 months. So these are old mice relative to what most people have been studying that are mice that are 3 to 6, 12 months old. And what we saw was a fairly rapid decline in the amount of the amyloid in the brain. These little red dots are amyloid plaques in both the cerebral cortex and in the hippocampus, and it was time dependent. The more we treated, the more we got rid of it. But it turns out that the amyloid not only occurs in these plaques, but it occurs around blood vessels. And you can kind of see some of those here, these little round circles that you see the red bits around. So this is a plaque, this is a blood vessel. They're pretty easy by eye to discriminate. So she decided to separately measure those two. And what she found was that while we were reducing by 90% the plaques, we were increasing by about threefold the amount of amyloid in the blood vessels. So we were busting up the plaque and it's going out along the outside of the blood vessels and reforming. And one of the things that had earlier been published was that this amyloid around the blood vessels can cause leakage of the blood vessels. Microhemorrhage is what we called it, what they call it. And so we decided to look, and sure enough, we saw the microhemorrhage. So these little blue dots up here in panel B, those are microhemorrhages where blood has leaked out of a blood vessel. And when we use the higher power microscope, you can see this vessel coming down into the brain from the surface of the brain with the blue around it. And in the adjacent section, this is the Congo red staining for the amyloid. So wherever we saw this leakage, we saw the Congo red. So somehow the antibodies are causing a local inflammatory reaction around the vessel, causing it to leak the blood into the brain itself. However, even with that, the mouse's memories came back. So you saw before that the mice at 15 months, they couldn't do the task. But what we found was that in the red line, these are the mice that got the control vaccine that have amyloid. In the black line are the amyloid mice that got the vaccine. The green and the blue lines are non-transgenic mice with and without the vaccine, and they all learned just fine too. So we were able to reverse the memory deficit in these mice. We were able to remove the pre-existing amyloid out of the brain. And that was really a very important observation. However, we got this hemorrhage issue. And none of it was serious in the mouse. We never had a big major stroke. But because of this, when the clinical trial started with the monoclonal antibodies in human beings, the FDA required them to look for this type of a problem to develop. And it was based on the research that had been done by the mouse people that they were even bothering to look for it. So it turns out in that vaccine trial that Elon had done, after 1 year, 2 years, 3 years, a number of the people in the phase 1 study had died, and they donated their brains and they went to autopsy. And what they found in the autopsy series that they obtained from people who were part of the vaccine trial was exactly what we saw in the mouse model. They saw that there was an increase in the amount of vascular amyloid, a big decrease in the amount of the parenchymal amyloid plaques, but they also saw an increase in these microhemorrhages. So that this was actually duplicating very nicely what we had found in those mouse models. And when they started putting back antibodies, monoclonal antibodies, into humans, and they started looking at the brains, they could find something that they called ARIA, amyloid-related imaging abnormality. And it's 2 forms. The first form was one that we'd seen. It's a microhemorrhage, and that you cannot really see it very well, but there's a little dark spot inside there that is the hemorrhage. But here, this bright spot is leakage of water out of the blood vessels. It's called edema. And you see both edema and hemorrhage often in the same person, not necessarily in the same place in response to this. However, most of the time, the person doesn't notice anything different. It's only observed because they're required to get the MRI. And if they end up having a number of the hemorrhages or they have severe edema, they're discontinued from treating with the antibody. The antibody levels drop down and it goes away. It's not an irreversible problem, but it is an issue, and Dr. Jaika will talk to you about that as we talk about rolling out these therapies. So the first drug that was used was the one with the very user-friendly name of bapineuzumab. If you remember that name at the end of the day, you do not have Alzheimer's disease. But bapineuzumab was a traditional antibody against amyloid, and it produced ARIA, and it produced other sorts of problems. And they were so concerned, they ended up decreasing the dose in study after study to the point that ultimately they're using 0.5 milligram per kilogram or 1 milligram per kilogram. Pay attention to that, because what I'm going to show you later is the drugs that work needed about 10 milligrams per kilogram. So this did not work. This is worsening cognition, and essentially there's absolutely no difference between the placebos and the 2 doses of bapineuzumab in terms of the worsening of cognitive function over time. The problem here is you got to treat for about 1.5 years before you can see enough change in the controls to see a benefit that you might get in your treated individual. This is another drug that was of the first generation of antibodies, a little better name, solanezumab. And in its phase 3 trials, it showed just a little bit of a benefit. So there's 2 ways that we look at cognition in older adults for these studies. One of them is to look at memory function, and one of them is to look at activities of daily living. Are they bathing? Are they brushing their teeth? Are they able to cook in the kitchen without causing problems? That sort of stuff. And in both the activities of daily living and in the cognitive memory work, you could see a slight difference, maybe a 5 to 10% slowing, but it wasn't statistically significant with this particular antibody. The interesting thing is neither of those antibodies appeared to actually reduce the amyloid in the brain under this circumstance. But the first antibody to do that is one that's called Aduhelm, also known as aducanumab in the initial, Aduhelm is the trade name. Aducanumab is a generic name. So we have these things called PET scans. A PET scan is a fairly simple procedure where you put a radioactive molecule on a chemical that sticks to the amyloid. And the radioactive molecule emits what's called a positron. That's what the P is in PET. And it's a very short-lived radioisotope. The amounts you get are very minor and modest. They're not going to harm you. It's just we use the radioactivity to detect where it is and how much of it is there. And so the PET scanner can detect it. And it's kind of like the radar maps for weather reports. You can see that where it's red, you've got lots of amyloid. And where it's blue and green, you've got less. So these are before and after pictures of the PET amyloid images from humans that got the antibody. So this is the placebo. You can see there's not much change over 1 year. But this is 3 milligrams per kilogram, 6 milligrams per kilogram. At 10 milligrams per kilogram, it's pretty much gone within a year. And in the phase 3 studies they had, you could see the amyloid going down in the ones that are treated in 2 different studies. And you can see that after they lose some of this amyloid, their cognitive function worse is up on this scale. Their cognitive function is decreasing at a slower pace. Unfortunately, these studies were terminated prematurely. And I don't completely understand how that happened. But because of that, they received what was called a conditional approval from the FDA based on these data. But Medicare refused to pay for it because it was still considered experimental. So Aduhelm is now doing another phase 3 study to see if they can't get a full approval to move forward with this treatment. If you looked at a second antibody, this one is called lecanemab, and the trade name for this is now Leqembi. It also was very effective at reducing the amount of amyloid on the PET scans to the point where the majority of people in this study were judged to be amyloid negative by PET scans at the end of the study. It had completely removed it from the brain. And they also showed about a 25 to 30% slowing in the rate of cognitive decline or activities of daily living in this study as well. Nonetheless, in both of these studies, the people were developing ARIAs. Most of the time, they're asymptomatic, a little bit less with Leqembi than it was with Aduhelm. And what the Aduhelm people did is they actually started to slowly ramp up the amount of antibody, and that decreased by about 50%, the amount of ARIA, the number of cases of ARIA that were observed by these groups. And so now there's a third one, and the third one is called donanemab. It doesn't have a trade name yet, but it probably will very soon. And this is from Lilly. And they also saw very rapid reductions in amyloid. And they also saw changes in other biomarkers that we have. So I told you about tau. Well, it turns out one of the things that happens in Alzheimer's is a phosphorylated form of tau builds up in your brains. And it can also be seen in blood. So we're doing blood tests now that can help us to diagnose who has amyloid and who doesn't. And this normally will increase in people who have Alzheimer's, but when you remove the amyloid, the phosphorylated tau also starts to go down. That's what the red line is here in panel C. And they saw a slowing of the decline in the activities of daily living, a slowing of decline in the memory function as well. So now we've got 3 antibodies, all of which are able to remove the amyloid from the brain. And all of them are slowing the rate of cognitive decline over a year and a half by about 30%. Now, the Aduhelm antibody was kind of in the lead at the beginning, and they have some cases that have gone 4 to 5 years of using these antibody treatments. Small groups, but what they're seeing is a flattening of the cognitive decline, not just a slowing. So it remains to be seen if that's ultimately what we're going to get. But it seems that you need to, first of all, really get rid of almost all of the amyloid before you start to see improvements or slowing of the rate of cognitive decline after the antibodies are developed. So the antibodies still cause ARIA, and several cases of people who have died now from complications of ARIA in combination with other agents. One woman had a stroke, and she was on the antibody treatment for dementia. And to treat the stroke, they use something called tissue plasminogen activator. It's a clot buster. And it dissolves the clots, but it also causes minor hemorrhages anyway. That combined with the ARIA caused a massive amount of hemorrhage in this poor woman. And she passed away very quickly after the tissue plasminogen activator was added. There's concerns about blood thinners as well, possibly also contributing to this type of an issue. So we're going to have to pay attention to this moving forward. But the antibodies do just slow cognitive decline. The problem may be that you've already lost a lot of neurons. Another part of the problem may be that you've got tau and it may continue to build up more slowly after the amyloid is gone. We don't know about that yet. But there is some suggestion that stabilization of cognitive decline after 4 years, and that's very encouraging. The problems are they're expensive. The treatment is probably going to be limited to very early stage cases. Severe dementia has not been tested with these antibodies, and it's unlikely that it's going to cause much benefit in those individuals, and probably may have even greater risks in those individuals. We may not be able to give the drug to all the people who might want to have it. But it's the first treatment that we showed benefited the mouse models of amyloidosis, and it is the first treatment to pass FDA muster as a drug to treat Alzheimer's. It just took a quarter century to get there. Amyloid is only one of the steps in Alzheimer's. And again, we've got that inflammation that Dr. Tanzi talked to you about developing drugs for. We've got the tauopathy that we're developing drugs for. And I think that in the future, we may actually get to the point that we've got safe vaccines. And people in their 50s will end up getting, along with their shingles vaccine and their flu vaccine and their hepatitis vaccine, they'll get a vaccine against amyloid. And maybe we won't hear about Alzheimer's. Thank you all very much.